Good afternoon, everybody. Welcome to the Northwest Illinois Agronomy Summit. I'm Philip Alberti, Commercial Ag Educator here in Unit 1. And today we are going to be welcoming Dr. Rodrigo Werle, who is the Cropping Systems Weed Specialist and Weed Scientist and Extension Specialist for the University of Wisconsin-Madison, my alma mater. And today he's going to ask the question, will the novel soybean herbicide tolerance traits solve my pigweed and ragweed problem? Dr. Werle is a specialist in the southern part of Wisconsin, but he does work all over, and I encourage you to take a look at the work that he and his team at Whisk Weeds do in University of Wisconsin-Madison, because quite frankly, sometimes the work that they're doing is more applicable to the producers in this area than some of the work being done in central and southern Illinois. I encourage you to type out any questions that you may have and be as interactive as you are comfortable with, but I'm going to pass this off to Dr. Werle to talk about his research and some of the strategies that producers can use to tolerate some of these problems. So with that, Rodrigo, thank you so much for being here today, and I'm going to kick things off to you to get this started.
All right, Philip, you have to stop sharing your screen so I can load up my screen. Yeah, awesome.
Sorry about that.
No, you're good. One sec here. Let me just. Can you see my screen?
Yes, we can. This is great.
Excellent. Alrighty. Thank you, Philip, for that introduction. Appreciate the opportunity to be here. About a year ago, we were all together in person. Wish could be that way. Hopefully soon here. But again, it's great for me to be here today, and I'll try to address this question that we have here. Will the novel soybean herbicide tolerance traits solve my pigweed and ragweed problem? And I think the question might be yes. For how long is a big question mark. Before I start here, I just want to thank you all for joining us. It's beautiful weather outside. The snow is starting to melt. So I know there's a lot going on outdoors. So I truly appreciate you being here for this hour with us. Okay, we got a small number of participants. So if we have questions, feel free to type them in the chat, or if you want to unmute yourself and ask questions, we don't need to keep the formalities that we typically do with large meetings. So again, if you have questions, if something doesn't make sense, just feel free to unmute yourself and ask a question. Okay, let's try to make this as informal as possible. So before I start my talk here, I just want to acknowledge our sponsors and our collaborators. As Philip indicated, we do a lot of research in southern Wisconsin at Arlington Ag Research Station, at Lancaster Ag Research Station, at our Rock County Farm by Janesville. And then we also been working at the O'Brien's Farm by Brooklyn, Wisconsin. So the O'Brien brothers, they let us use every year about 5 acres of their production fields where they have severe infestation of waterhemp. And this is where we've been doing a lot of our waterhemp research. So we're very thankful to our collaborators, our sponsors, the commodity boards, the United Soybean Board, the Wisconsin Soybean Marketing Board, and the Wisconsin Corn Promotion Board for sponsoring our efforts, and also our crop protection industry. And then lastly here, I would like to take a minute to acknowledge this amazing group of individuals whom I have the opportunity to work with on a daily basis. This is our research lab here over the past 3 years since we started here at UW-Madison in January of 2018. And I just want to point out that Dan Smith here, he's with the NPM program. He gave a talk about cover crops a couple weeks ago, and we have the pleasure of working with Dan pretty much on a daily basis as well. So for my presentation here today, I'm just briefly going to go through some of the work that we've done looking at giant ragweed and waterhemp biology. We'll talk about what's going on in regards to herbicide resistance here in the state of Wisconsin. And then we'll talk about some of the herbicide selection work that we've done looking at pre-herbicides, burn down, but also systems, okay, or integrated weed management approaches. And then lastly, I'm going to wrap this up talking about some of our cereal rye cover crop research. I know we've been talking about cereal rye cover crop for a while now. Some producers are becoming more and more interested, and we'll provide some of our data here and share what we've been learning over this past couple of years in regard to that topic. So 2021 growing season is here already. Pretty soon we're going to be hitting the ground and planting. I think most farmers have made their decisions in regard to which platform or which varieties they're going to be planting. There's been a lot of innovation coming down the pipeline over the past few years here, but most of them are not, like new chemicals. Pretty much, the new GMO events that we have in our soybean crop. So now we can use more of our, if you would, our corn herbicides into our soybeans, giving the farmers more options for post-emergence control of some of the troublesome weeds we're dealing with. So just an example here, the Extend platform conferring tolerance to dicamba post-emergence, and List platform, which is becoming quite popular here in Wisconsin from recent conversation with our producers. The reason being is that farmers can either use glyphosate or 2,4-D choline post-emergence, 2,4-D choline being somewhat simpler to spray than dicamba. Okay, so that's one of the reasons why some of the farmers are going that route. However, recently we had the introduction now and the commercial launch of the Extend Flex platform, okay, which confers tolerance to glyphosate, dicamba, and glufosinate post-emergence. So bringing a little bit more flexibility to farmers that like the Extend platform, but are rather not use dicamba. Now they can use glufosinate post-emergence. Since we're on this topic here, one thing I want to urge our farmers to keep in mind is that, glufosinate is this common herbicide across our platforms, our Enlist, our LGT27, and our Extend Flex. So, I do believe we're going to be putting a lot of selection pressure in glyphosate. Glyphosate can be a very effective herbicide, but if we don't use it properly, I think we might, we'll start running across resistance issues pretty soon. And this is actually what's happening down south already. And this is going to help me address my question. Will these platforms solve the problems? Yeah, they might, short term. If we don't use them wisely, we're going to have problems. So about a couple of weeks ago, this news came out. And I'll be honest with you, I was upset, a little sad to hear about it. So our colleagues down at the University of Arkansas, they have reported the first Palmer amaranth population resistant to glufosinate, okay? And they found this population in cotton fields where glufosinate has been used for multiple years, multiple times within a growing season. And this is what we got. So here, just showing the population here being treated. I'm sorry, I'm trying to figure out the cursor thing here. One sec. So this is the population here that's confirmed to be resistant. This is just like a 1X application of Liberty, 32 fluid ounces. Okay, and as you can see here, not doing the job controlling this Palmer amaranth post-emergence. So that's very unfortunate. This is just what came down recently. Last year, we also heard of reports out of Tennessee, Palmer amaranth that was confirmed to be resistant to dicamba. We have Palmer amaranth in Kansas resistant to 2,4-D and dicamba. We have waterhemp in Illinois. We have waterhemp in Nebraska, and we have waterhemp down in Missouri confirmed to be resistant to 2,4-D. Okay, so what's my point? If we don't use these three herbicides wisely post-emergence in soybeans, pretty soon we're going to start to see this coming our way. And one thing that I want to point out is that Palmer amaranth and waterhemp are closely related. They're cousin weed species, if you would. They are in the same plant family. So what happens to Palmer is likely to also happen with waterhemp, and we need to keep that in mind. The other reason why that concerns me, particularly here in Wisconsin, the systems that I work with, our dairy producers, they bring a lot of cottonseed from down south. And I just, Palmer amaranth is a major problem down there. Resistance, it's happening, new cases after new cases all the time. So this is a concern that we have as we bring this cottonseed to our dairy industry up here in Wisconsin. We have this opportunity of bringing Palmer amaranth seed. And this here are pictures from two encounters that we had last year with Palmer amaranth. This one here in southwest Wisconsin, and this one at Arlington and Columbia County in south central Wisconsin. This is a private operation. We visited, we confirmed Palmer in the corn production. We took a walk around, and at the end of the day, the farmer, shared with us that he was using cottonseed and he had no idea of this linkage of cottonseed with Palmer amaranth. So soil residual herbicides are the way to go with waterhemp. Effective burn down herbicides are the primary way to go with giant ragweed. The other thing that I want to point out is that water hemp seeds will last for several years in the seed bank, 6, 7, 8, 9 years. Whereas giant ragweed, it only lasts a couple years, okay? So water hemp will require long-term management, whereas if you do a good job for a couple years with giant ragweed, you can crash that population. Okay, so now I'm gonna start talking about resistance and what's going on. So resistance to group 2 and resistance to group 9 is widespread in both water hemp and giant ragweed. Here's a slide that I have for ragweed, just demonstrating that group 2 and group 9 resistance, or ALS resistance and glyphosate resistance, have been confirmed in 7 and 12 states respectively, so it's widespread, including here in Wisconsin. We then have a graduate student named Felipe Faleco, and Felipe's effort is being sponsored by the Wisconsin Soybean Marketing Board. He's doing a lot of screenings in our greenhouse. So in the fall of 18 and again the fall of 19, we got some populations from southern Wisconsin, giant ragweed, and they were primarily coming from systems where non-GMO soybeans are raised. Okay, so these are primarily coming from non-GMO soybeans where the growers are really struggling to control water hemp. So what we did, not water hemp, giant ragweed, I'm sorry. So what we did here, we had 3 treatments: first rate or chloranthalum, glyphosate, Roundup Power Max, and then Flagstar or Fomesafen. So these were the 3 treatments we looked at. And then we had this rate structure. We had a 1X or a commercial rate that you're all using. And we also tested them with a 3X, a very high rate, to see if there's different levels of potential levels of resistance in these populations. So this is how we do these studies. We let these plants grow when they're about 2 to 3 inches in height. We spray them with our treatments, and then Felipe comes and collects visual assessments and also biomass. And here's what their visual assessments look like. So here I have a plant that's completely controlled, and these are the plants that concern us. Okay, this is 21 days after spraying that post-emergence herbicide. So these plants here, we rank them as concerning plants or putative resistant plants in our populations. So the overall results from our giant ragweed efforts, 9 populations being tested. What we learned here, if you look at the 1X, is that 3 out of 9 populations were classified as resistant to group 2 herbicide. We had 2 glyphosate resistant giant ragweed populations in our screenings. And then this is the one that's really getting our attention here, okay, which is this one that we confirmed to be putative resistant to PPO herbicide or Fomesafen. We went into the literature. No reports of PPO resistance in giant ragweeds have been confirmed yet. So we're going to continue to do some work here. And this is really concerning because the fields where these populations are coming from, they're primarily treated with Fomesafen or Flagstar post-emergence. That's one of the primary options that these non-GMO growers have for giant ragweed control. So this is, again, really, really concerning. And it's just an indicator that if we use the same chemistry time after time after time, eventually this weed species, they will evolve resistance. And here is just showing pictures. So this is one of the populations that Felipe classified as ALS resistant. Oops. Here is the untreated plant 21 days after. This is what with the 1X, a first rate, and this is the 3X. You see the plant surviving, doing just fine. Here's an example of a glyphosate resistant population, untreated control, a 22 fluid ounce of Roundup, here not controlling this plant. And this is this population of concern that we have from Rock County, not too far from Northern Illinois, where this 1X rate of Fomesafen is not doing the job. So this 1X rate of Fomesafen controlled all 8 populations, but there's one population here, again, very concerning, and we're going to continue to do some work. Okay, are there any questions or concerns to this point? Cool. Alrighty, so now I want to switch gears and I want to talk a little bit about water hemp here. Okay, so the light green here indicates resistance to group 2 herbicides. So these are the counties in Wisconsin where we have known resistance to group 2, and imazethapyr is the herbicide that we've been using for our screening. And as you can see, it's widespread. We know it goes to more counties than what we have reported, but this is the data that we have. When we test these populations in the greenhouse, what Felipe has found is that more than 95% of these water hemp populations are ALS resistant. The map in the middle here in red are the counties with confirmed glyphosate resistance. That's not surprising to you folks in Illinois. If you've attended the talk from Dr. Aaron Hager there out of the University of Illinois, he often shows us maps, and you know that glyphosate resistance is all over Illinois, and it's now all over Wisconsin. This last map here is PPO resistance, or resistance to group 14, and this is something really interesting here, because when I started in the job, I inherited the map from my colleagues in the Department of Agronomy. And when I first presented this map in January of 2018, there were 4 counties in Wisconsin where we had known PPO resistance confirmed, okay? In the fall of 18, we updated this map. And then in the fall of 18, we had 10 counties with known PPO resistance. We have just updated this map in December, and now we have 22 counties in Wisconsin with confirmed PPO resistance. So as our farmers are struggling with ALS and glyphosate resistance, they switch into PPO herbicides pre and post. And this is what we're seeing here, this increased level of PPO resistance. And I want to point out that these are samples from Wisconsin that we sent to the plant clinic lab out of the University of Illinois, and they look for the PPO mutation. Okay, so this is how we generate these maps. Here's a brief summary of what Felipe has been doing in the fall of 18. We put a call out for our producers and agronomists and educators all around the state. We asked them to collect water hemp seed and send it to the university, okay, to campus in Madison. And for this past 2 years, Felipe has been screening them with several pre and post herbicides. What you're going to see in this map here are a bunch of golden dots, and that summarizes what I was just saying. The golden dots are those populations that have resistance to glyphosate and imazethapyr, so 2A resistance quite widespread. Here in the western part of Wisconsin, where we use a little more atrazine, where we don't have as many restriction zones as we do in the southern part of the state and central part of the state, we're starting to see this 3-way resistance of glyphosate, imazethapyr, and atrazine. And then one population that really got our attention is this one here from Fond du Lac County. And this population here is confirmed to be resistant to glyphosate, imazethapyr, and we suspect that 2,4-D is not working in this population. And what's really interesting here is that 2,4-D, we just went back to the records that the farmer sent us, and 2,4-D hasn't been used in this operation, okay? So it's really intriguing that we're seeing 2,4-D resistance in a population where 2,4-D that hasn't been exposed to 2,4-D. And here are the pictures from this population. So the control plants, 21 days after application, oh goodness, 21 days after application, this is the response to imazethapyr, not working, glyphosate. Even though we didn't classify it as atrazine, we had some plants that escaped our atrazine treatment, and here's the 2,4-D. The only herbicides that worked in this population were glufosinate, fomesafen, and mesotrione. And across our trials in the greenhouse, the only herbicide that provides 100% control of all populations is glufosinate, or Liberty. Okay, and that goes back to the reason why I say we need to be good stewards of this herbicide. So this is some of the resources that are available to producers and the agronomists and educators out there. If you don't have these charts, please, I urge you to go and Google Take Action Herbicide Mode of Action chart. This is a very, very valuable resource for farmers and agronomists developing their herbicide programs to make sure that they are using multiple effective herbicides. Okay, so I just want to put a plug for that. Alrighty, so talked about resistance, talked about the 2 primary weeds. Now I want to start talking about developing your weed control program for 2021. And I believe at this point, most of you have the decisions made already. So either I want to present some data that will reinforce your decision or may help you fine tune it before we hit the growing season. Okay, so for soybeans and also for corn, it is important that we start clean, whether that's through an effective burn down in no-till systems or tillage in conventional tillage systems. And at that point, when we're establishing the crop, it's important that we put down an effective pre-emerge herbicide program. Picture on the left is a scenario 25 days after planting where no pre was sprayed. The picture on the right is where you have a pre, okay, an effective pre was sprayed at planting, and were we in the same room, I would be asking you right now, where would you rather be? Okay, so there are several benefits to this scenario here. I know it comes with a cost, the cost of the product and the application, but there are several benefits. Some of them, you know, an effective pre-emerge herbicide is independent of herbicide tolerance trait of choice. So you can use the same pre-herbicide in Xtend beans, Enlist beans, Roundup Ready beans, Liberty Link GT beans, and in non-GMO beans. Okay, so that's one of the beauty about pre-emerge herbicides. They protect your crop from early season yield loss. At this point here, you're already losing yield. The weeds are already taking yield away from you. Here you're protecting your maximum yield potential. They're buying you time for a post-application. Some of these plants are already passing that 4-inch mark. And if you start getting rain at this point, trying to control this post-emergence is gonna be a nightmare. And then they're also reducing the number of plants. And this is very important. When you put a good pre down, you're reducing the number of plants that need to be controlled post-emergence, further postponing resistance evolution to the post-emergence herbicides that we have available to us. Okay, so this is some studies that we've been doing since I started here at UW-Madison, 3 years of data now. I'm gonna present a data set here from Brooklyn, Wisconsin, 2019, looking at water hemp control on the left. And then I'm gonna present some 2020 data looking at giant ragweed control on the right here. So for these trials here, we call them the Whisk Weeds Management Challenge, the comparison of pre-herbicides. So what we do, we plant our beans, we spray this 30 different herbicides, and then at 25 and 50 days after application, we evaluate residual weed control. So the idea here is to help our farmers select effective pre-emerge herbicide programs that work well, not only for water hemp, but some other weeds that we have in our seed bank, such as giant ragweed. Okay, so I want to acknowledge Ryan Worth, a research specialist in my program, for generating this slide here. And the way we have it separated, you're going to see a bunch of colors. So from Zedra upwards here, these are herbicides with one side of action. So only one active ingredient, one side of action. We have group 2 herbicides. We have a group 5 herbicide, Metabusing. We have three group 14 herbicides. And then here we have our group 15 herbicides. The red bars here are premixes containing 2 sites of action. And the yellow bars are premixes, commercial premixes containing 3 sites of action. And as you can see here for water hemp already, as you start using these premixes with 2 or more sites of action, you're already enhancing control. We want the control to be as close or above 90%, okay, to consider that an effective program. So let me take a step back here. Again, we're gonna do some comparisons of water hemp on the left and giant ragweed on the right. So ALS herbicides, they don't work quite well from a pre-emerge standpoint in water hemp, less than 50% control. However, they're still effective for giant ragweed. Group 2 herbicides are the foundation for residual control of giant ragweed. Okay, so a lot of folks are removing group 2 herbicides from their pre-emerge program because they think there's no value to group 2s. I just want to warn you that the group 2 herbicides still bring a lot of value. By themselves, they don't work. But if you combine them with something else, you can get pretty good residual weed control, and that's what we see here. Okay, another one here is a good one is Metabusing. A lot of producers are bringing Metabusing as part of their pre-program. Metabusing is quite effective for water hemp, not as much for giant ragweed. Okay, so again, understanding what you have in the seed bank will help you decide the best way to proceed with your pre-program. One thing that I want to point out is that this pre-mixes of a group 14 and a group 2 did quite well for both ragweed and water hemp. Okay, and then one example that I like to pick when talking about this is Boundary, for instance. So Boundary, it's a pre-mix of Duo and Metabusing, so a group 15 and a group 5, excellent water hemp control. But then you come over here and you look at the giant ragweed, it's very poor control. Again, a group 15 and a group 5. Here's the data for Duo on giant ragweed. Here's the data for Metabusing on giant ragweed. And as you can see, there's 2 AIs that are not very effective on giant ragweed. However, they are extremely effective for water hemp. So combining them, excellent water hemp control, not necessarily good giant ragweed control. Okay, so again, understand what the different modes of action, the value that they bring to your system. And I can make the slides available if you're interested. Now I want to switch gears in a similar story here. It's for corn. So this is a study here that we conducted for corn. We compared 30 different herbicides. These corn studies are conducted a little bit different. What we do, we come and we spray our herbicides at the V2 growth stage, simulating this common scenario that we have here in Wisconsin, where our farmers work the ground, they plant the corn, and then they come with a one-pass herbicide program early post. So we had glyphosate or Liberty as a tank mix because we didn't want to evaluate burn down. We only wanted to evaluate soil residual control. So all these herbicides here, they were tank mixed with glyphosate or Liberty at 32 fluid ounces. So again, we're only evaluating residual control data presented here 25 days after that treatment when the corn was about V6, V7. And a similar story that we observed for the soybean study, we're observing for the corn study. When you use pre-mixes, you're more likely to enhance residual control of water hemp. The one thing that we notice in our corn trials at Rock County Farm in Janesville is that this one-pass system does not do a good job for giant ragweed. Remember what I was saying early on, that giant ragweed, large seeded, relying on soil residual for giant ragweed control, not looking very promising. The best herbicides there were actually Verdict at the 15 fluid ounce rate. Remember, for soybeans, we use 5 ounce. For corn, we use 15 fluid ounce, a little higher rate there. So it did quite well. For ragweed, followed by some of the Harness, Acheron, and Razicor there. Okay, so there are some good products that work just excellent for water hemp, but the residual activity is not as strong for ragweed. So for ragweed, our farmers have to put a pre, have to scout, and come back with a burn down if they're dealing with severe infestations. So to this point, I presented residual weed control, and that was a major effort from our program to help growers pick the right pre-herbicide. Once we started sharing those data with our growers, their question was, okay, Rodrigo, now I know which pre-herbicide to use. How about post? I'm in season. I want to do a burn down application in season to control weeds that escaped the pre-emerge herbicide application. What should I use? So in order to address that question, we put this bare ground trial. So we worked the ground with tillage. We let the weeds come, and then when they're about 4 inches in height, we spray them with 20 different post-emerge herbicides for several sites of action, as you're going to see the sites of action numbers here. On the left, again, same type of slide, water hemp on the left, giant ragweed on the right. The first thing you're going to notice is that you have a lot of bars going above 90% for giant ragweed. Going back to my initial point, ragweed is easier to control post-emergence, whereas water hemp, it's a challenge to control post-emergence. So the best way to go with water hemp control is through soil residual herbicides. Thinking about a soybean system, let's discuss the most effective post options for water hemp. The first one in our bare ground trials was glyphosate, about 95% control, followed by Enlist 1, 2,4-D-choline, followed by Extended Max or dicamba. So as you can see, there's new platforms, there's new herbicides that we can use in our traded crops. They bring a lot of value from a post-emergence standpoint. It's also not uncommon for me in June, to start getting calls from our growers asking me, Rodrigo, I have severe water hemp. I'm going to go with a PPO post-emergence. Which one should I use? So to address that question, we did this comparison here. We have Resource, Cadet, Flagstar, and Cobra. And in our trials, Cobra and Flagstar, they tend to provide similar performance, about 80% control, followed by Resource, and then Cadet. Okay, so Cadet is not the right PPO nor Resource for burn down of water hemp. Cobra and Flagstar tend to do a little better. And the similar trend we observe for giant ragweed here, Cobra, Flagstar, followed by Resource and Cadet, glyphosate and dicamba, 2,4-D also doing an excellent job for giant ragweed post-emergence. So again, a lot more options for giant ragweed post-emergence burn down than for water hemp. Okay, So this goes back to this 1 versus 2 pass system, and particularly in corn, this discussion that we have with growers a lot. I'm gonna I'll ask you to pay attention to this 3 bars here. So Ryan Dwarf in our program, we had about 20 trials down at Rock County Farm. And then what he did, he compiled all those trials and he ran some averages here for us. So the figure on the left is giant ragweed control, and I want to talk about this end of season data. If you go with a 1 pass spraying the herbicide at the time of planting, at the end of the season, we had about 65% control of our giant ragweed, severe infestation of giant ragweed at this site, by the way, and the error was big. If you go with a 1 pass early post at V2, V3 growth stage, the control was slightly better, about 75%. Now, if you go with a 2 pass system where you have the severe infestations, this is when control was optimized. Okay, I know that comes with a cost, a 2 pass program, but that was optimized. Now is grain yield. If you don't have an effective herbicide program there, the average yield of our untreated check plots was about 60 bushels to the acre. Put in a pre-herbicide only at planting, we harvest about 200 bushels. If we move that 1 pass application from the time of planting to early post, we saw a 50%, or not a 50%, a 50 bushel yield increase here, okay? The one thing that I want to discuss here now is that, okay, a 1 pass early post versus a 2 pass, the yield was about the same. So we didn't see much of a difference, with the late weed escapes. For me as a weed scientist, the problem that I see with that, okay, you might not have an immediate return on the investment here with a 2 pass, but if you don't go that route, if you let the weed escapes, even though they're not impacting your grain yield, this escapes will produce weed seed. You go back next year with soybean, you're gonna have a heck of a time controlling those plants. And that's why I make this strong case here to take advantage of the herbicide programs that we have available for corn. Okay, so just some of the conclusions here, because of widespread resistance, primarily glyphosate, but also some other herbicides, that really complicates post-emergence weed control. Therefore, we need to use this integrated systems, looking at effective burn down tillage, picking good pre-emerge herbicide options, and being wise with the post-emergence herbicide selection. All right, so now I talked about enough chemistry. I want to switch gears a little bit and talk about integrated weed management. We've been doing this work now for a couple of years, so we got some neat data to share with you all. So this here, what you see here in this slide in front of you are pictures of our soybean plots at Arlington Ag Research Station. The pictures on the left, 30-inch row spacing. The picture on the right is a 15-inch row spacing scenario. Same seeding rate, same planting date. And then the question that I have for you, if you were a weed, let's say a waterhemp or a giant ragweed plant trying to emerge and grow, where would you rather be? In this scenario here or right here? So this is picture from the top. This is picture from down below what the weeds would be seen, right? So where would you rather be? So in order to address that question, we had a graduate student actually investigating this factor. So what he did, he transplanted small waterhemp seedlings every week throughout the growing season to see the impact that Canopy Closure has on waterhemp growth, okay? So these are, I'm gonna point out the seedlings that were transplanted on June 14th, and we harvested them at the time this plant's flowered. Here's when there is no crop competing. So you have this bushy plant or this Christmas tree, if you would. Here is in a scenario where you have 15-inch row spacing soybeans, 30-inch row spacing soybeans, 30-inch row spacing corn. Here you see the impact that a 15-inch, the narrow row space and the rapid canopy closure is having on waterhemp. It's buying you time for the post-application. And if this plant here just so happens to escape control, the amount of seed it will produce is significantly lower than the amount of seeds this plant here would have produced in a 30-inch system. With that in mind, we conducted this, what we call this agronomic trial, trying to address a question that we get from producers in regards to early season crop injury from pre-emerge herbicides, okay? So what we did, we investigated planting time. So we had 2 different planting times. We have what we call for us here in southern Wisconsin, very early planting, which was April 25th in 19, and then April 21st in 2020. And then we had what we call our standard planting time, which is May 23rd and May 22nd in 19 and 2020 respectively. So again, the first factor in our study was planting time, early versus standard. The second factor that we investigated was row spacing. We had 15-inch row spacing soybean, and we had 30-inch row spacing soybean. We had tillage. We compared no-till with conventional tillage. And then the last factor here again was trying to address the grower's question, a yes pre-herbicide and a no pre-herbicide. And our pre-herbicide of choice was Fierce MTZ, which contains Metabusing, pyroxasulfone or Zidua, and flumioxazin or Valor. So this three-way combination is known to cause early season crop injury when the conditions are not adequate. So especially when you have cold and wet soils, this is when we tend to see more injury from this particular pre-emerge herbicide. So we wanted to understand how these factors impacted canopy closure and soybean grain yield. One of the data that our student responsible for the study, Nikola Arsinovich, collected was canopy closure. So through his phone, there is an app that's called Canopio, where you can track ground coverage. And this is what you see in this picture here. He will take an image. So this is the raw image that he took with his phone. And then here is a processed image with Canopio. So it tells you how much green canopy you have within that image. So this particular picture here, he had about 18% green canopy. For his study, we wanted to understand the time that it took for soybeans to completely close canopy or reach the 90% canopy closure, which is the point where he is at here in those plots right now. The one thing that I want to point out, we had one study in 19 and one study in 2020. If you recall, 2019, we had a lot of precipitation that year, whereas in 2020 was more of a normal year, if you would. It was warmer earlier, but then in August was a little bit drier. So the weather played a major role in what we observed. And the other thing is that the 19 study was done in a field where we had continuous corn prior, and the 2020 study was conducted more in a corn-soybean rotation. Here are the main findings from Nicola's study. The first one is the time to canopy closure. We have a lot of response variables, so for sake of time, I'm just going to provide the results of what we call this random forest. So we look at the entire data set, and then we ask this random forest, what are the factors that are driving canopy closure? And then we rank them. So the most important one was year. The year played a major role, so the soil conditions plus rainfall and temperature was quite different between the 2 years. So that impacted canopy closure. The next factor that had a big impact, something that we or farmers can control, was planting time. The sooner we planted the beans, the faster they close canopy. And this is what we want to see. And that's what the soybean agronomists all over the country tell us. Let's go plant as soon as possible, because the sooner you plant, the more likely you are to optimize grain yield. So same idea here with canopy closure. The next factor was row spacing. So going 15-inch space had a tremendous impact on how fast that canopy closed. And again, I showed you a picture already showcasing that. And then next was tillage and herbicides. These two practices here, they did have an impact, okay? But what's most importantly here, even though we did see some early season injury from that pre-herbicide, it wasn't a major factor impacting canopy closure. Now I want to switch gears and I want to talk about grain yield. Big difference in year. In 2019, our average in our study here was about 95 bushel of soybean, whereas in 2020 it was a 60 bushel. And that's why year is playing a major role. The next was planting time. The sooner we planted, the higher the grain yield. Tillage had a little bit of an impact. In our case here, our tillage plots yielded a little more than the no-till. And then lastly here, herbicide and row spacing had no impact. So we were happy to see these results because pre-herbicides, they had a little bit of impact on canopy closure, but they had no impact whatsoever in our study on final grain yield. Okay, so that's great news. Plus all the benefits that pre brings to our system that I just described in regards to weed control. The next thing here is that even though row spacing helped with faster canopy closure in our trials in southern Wisconsin, we did not observe an increased soybean yield with that by going narrow. Okay, So several studies have been conducted. Going narrow not always necessarily lead to higher grain yield. And here's just the take home from Nicholas' trial here. Earlier planted soybeans, faster canopy closure, higher grain yield. Narrow space soybeans, in our study, we observed faster canopy closure, but we did not see a yield gain compared to the 30-inch system. Pre-herbicides, it slowed canopy closure a little bit and had no impact on grain yield. And conventional tillage, a little faster canopy closure and a little higher grain yield. Okay, I'm coming close to the end of my presentation. I want to continue to talk just a little bit more about integrated weed management. A couple weeks ago, I had a chance to attend the annual Weed Science Society of America meetings. And, you know, from folks working down in Georgia all the way to folks working up north here in Wisconsin, one strategy that we're seeing really promising results is what we call this planting green, okay, into standing cereal rye, terminating that rye and combining that high biomass system with an effective pre-emerge herbicide program containing 2 sites of action. This is when we're seeing the best levels of weed control. We have a graduate student. I just want to point out here, we just received some funding through the United Soybean Board. As we speak here, we have an ongoing study all over the country, 11 universities, 12 locations investigating the impact of planting green on weed control and soybeans. So stay tuned for that. Hopefully, Philip, if you invite me back next year, we can talk about this study here. I have a graduate student here in Wisconsin. For the past 2 growing seasons, he's been investigating the impact of soil management strategies on soybean yield. Here we have 2 years of data summarized from Arlington, 2 years of data combined here for Lancaster, so 19 and 2020, where we look at different soil management strategies. So we have tillage, you know, as a conventional system, a no-till, cereal rye terminated 15 days before planting, which is kind of a standard recommendation. I don't like that very much because if you're killing your rye 15 days before planting, that rye is going to decompose really fast. It doesn't bring weed control benefits. This is terminating at planting, the day you plant. This is chopping for silage, and this is terminating that rye 15 days after. What I want to point out here is that soybeans are quite resilient, and regardless of the soil management strategy that we adopted, we did not see impact on grain yield. I personally really like this strategy, again, letting that rye accumulate as much biomass as possible and then terminating it, especially here if in our region we have enough precipitation allowing for that type of system. And this is just showing a picture here of what that green planting looked like. So this is the 3rd week of May. We have our crew here establishing Kobe's plots. So just going and planting green into the standing cereal rye and then terminating that rye afterwards. Really promising system. Dan talked about this a couple of weeks ago, Dan Smith from the NPM program. Another integrated strategy is prevent the movement of weed seed, whether it's through equipment, whether it's through dairy feed, whatever that is, whatever we can do to control, we need to take advantage of such opportunities. And here's what we do in our combine clinics. We go around and we talk about the importance of not letting pigweeds go to seed. And if they do go to seed, make sure you clean your combine as you go from field to field so you're not bouncing those seeds around. So for the future of our program here, where we're headed, we're looking into this system here. We've been doing a lot of work looking at water hemp suppression. The question that we get is, will this system also work for giant ragweed? Okay, how does biomass, does that suppress giant ragweed? We don't know. We see a lot of giant ragweed growing through wheat. So we don't think this high biomass system will be as effective for ragweed as it is for water hemp. So we're studying that. We're going to continue to do some work looking at the PPO resistant giant ragweed population I talked about, and that putative 2,4-D resistant water hemp. So we're going to continue those projects. And this is an area we're diving in. We talk about this. Some people don't like it. Some people like it a lot. We started doing some drift trials with hooded sprayers. So that hood that goes over the top of the crop, the broadcast hood, if you would. We saw promising results reducing drift. Now what we want to do is look at these types of hoods here, see if we can bring different chemistry into our weed control programs, bring in some of additional herbicides that we couldn't use otherwise. But if we're protecting the crop, if we're only spraying in between the soybean rows, how can we use the system? So we've purchased this equipment and we're excited to start exploring it this growing season. Wrapping up, lots of chemistries. Resistance is a concern. We have some effective options. Post-emergence, we got to use them wisely, especially this, the dicambas, the 2,4-Ds, and the glufosinate that I've been talking about in our soybean systems. We need to be good stewards. And whenever possible, I like to urge our growers to implement at least 2 non-chemical strategies, or kind of think outside the jug. And just today here, I provided 3 different examples, stopping the spread of weed seed, using high biomass cover crop systems, or adjusting some of the agronomics or planting time and row spacing to promote canopy closure. Everything that I presented on today is available on our website. Philip already mentioned, but if you go to whiskweeds.info and then you click the reports tab, everything is there. So we just released our 22 Whiskweeds report. We have a lot of pictures from our plots that we just made available to you all. So you can click these various links here, download the PDF is there for you, and hopefully is of, you can put this files to good use. With that, I want to thank Nick Arneson, who helped me develop this presentation. Nick is the outreach specialist in my program. We got about 10 minutes here until 2 o'clock. If we have questions, I think this will be the time for it. Philip, thanks again for the opportunity. I hope the information presented today was helpful. Here's my email, and if you're on Twitter, make sure you follow us there. This is where we push out most of our updates. So thank you all very much.